Hello, good afternoon, everybody. Can you hear me? Hello.
Good afternoon, sir.
Okay. Now more than 70 are present. Now we'll start. Okay. Can you see my slides?
Yes, sir, it
Yes.
is visible.
Okay. Now we'll start.
Yes, sir.
Okay. We're studying about fatty acid metabolism, and in our earlier classes we studied about the breakdown of fatty acid or the oxidation of fatty acids, how energy is generated from the breakdown of fatty acid or the oxidation of fatty acids. And with that, we studied giving importance to beta oxidation. And in beta oxidation, what is the final product? Acetyl coenzyme A. And when there is the large amount of beta oxidation, large amount of acetyl coenzyme A would be there, and that acetyl coenzyme A will lead to the formation of ketone bodies. And we also studied about the synthesis and utilization of the ketone bodies. Now today we will study about fatty acid synthesis and triacylglyceride synthesis. How fatty acids are synthesized. We have already oxidized the fatty acids, isn't it? And calculated the energy released from the oxidation of fatty acid. Now we are going to study how fatty acid is synthesized in our body. And after fatty acids are synthesized, the fatty acids are stored in the form of triacylglyceride. And in the next class, we'll cover about triacylglyceride synthesis also. Okay. Fatty acid and triacylglyceride metabolism. This is the syllabus. This is our syllabus. You must already be aware of this syllabus. Syllabus we might have already seen. Okay, aspects on fatty acid synthesis, tissue precursor, overview of conversion of acetyl coenzyme to fatty acid, and in this also we'll take the example of palmitic acid, and we'll also look at the regulatory mechanism. After that, mechanism of conversion of palmitic acid to next higher saturated or unsaturated fatty acids, because during the synthesis of fatty acids by the main mechanism which we are going to study, this will lead to the synthesis of palmitic acid. And if other unsaturated fatty acids have to be synthesized, then only after the synthesis of palmitic acid there will be the insertion of the double bonds, or there will be the elongation of that palmitic acid. Biochemical basis of nutritionally essential fatty acid. You have already studied about the nutritionally essential and non-essential fatty acids. The definition is similar to that of essential and non-essential amino acids. But please don't get confused with the nutritional classification of proteins. In the nutritional classification of proteins, you have studied about complete protein, partially complete protein, and incomplete protein. That is the nutritional classification of protein. However, the nutritional classification of amino acid and the nutritional classification of fatty acids are essential and non-essential. And after that, we'll study about the triacylglyceride formation and storage. Okay, this part I have already gone through. Aspects and beta oxidation of fatty acid, palmitic acid, acetyl coenzyme, tissues, and energy are most important part. That's why I have already covered. And at last, we'll also see the hormonal intervention in the reciprocal regulation of lipogenesis and lipolysis. Okay. Now, synthesis of fatty acid. A large proportion of the fatty acids used by our body supplied by the diet. However, de novo synthesis also do occur and mainly occur in the cytoplasm of liver, kidney, adipose tissue, and lactating mammary glands. We have studied about the beta oxidation, that is the oxidation of fatty acids, that we have studied in the mitochondria. Now, de novo synthesis occurs in the cytoplasm of these tissues, liver, kidney, adipose tissue, and lactating mammary glands. And these are the requirements for the synthesis of fatty acids. We have seen that after the complete oxidation of fatty acids, acetyl coenzyme were produced. Similarly, for the formation of fatty acids also acetyl coenzyme are required. They are the first and foremost requirements. Acetyl coenzyme as a source of carbon, and we also need NADPH as a reducing equivalent. This is the synthetic pathway, and we also require the reducing equivalent, and we require NADPH as a reducing equivalent. And we also require energy. That energy is in the form of ATP. That means the requirement for fatty acid synthesis are acetyl coenzyme as a source of carbon, NADPH as a reducing equivalent, and adenosine triphosphate, that is ATP, for the energy supply. Synthesis of fatty acids requirements, three requirements: acetyl coenzyme A, NADPH, and ATP. Okay. Synthesis mostly occur in liver, kidney, adipose tissue, and lactating mammary gland. Okay, now we'll see the brief outline of fatty acid synthesis. In beta oxidation also, we have said that the beta oxidation will occur in three phases or three stages, like activation of fatty acid occurring in the cytosol, transfer of that activated fatty acids to the mitochondria, and beta oxidation proper in the mitochondria matrix. That was the brief outline of beta oxidation. Similarly, here the brief outline of fatty acid synthesis. First of all, the production of cytosolic acetyl coenzyme and NADPH, because here fatty acid will be synthesized in the cytoplasm. That means we require the acetyl coenzyme A in the cytoplasm. Why we are focusing production of acetyl coenzyme A and NADPH in the cytoplasm? Because acetyl coenzyme A is formed in the mitochondria. Now that acetyl coenzyme A, which is formed in the mitochondria, either through the process of conversion of pyruvate to acetyl coenzyme A, which follows that glycolysis, or from the breakdown of fatty acids, or from breakdown of amino acids. So the metabolism of amino acids, that acetyl coenzyme A is formed in the mitochondria. Now that acetyl coenzyme A, which is formed in the mitochondria, has to be transferred to the cytoplasm. So the first step is production of cytosolic acetyl coenzyme A and NADPH. You know the major source of NADPH is the HMP shunt, hexose monophosphate shunt. Okay. Now after this, there should be the conversion of acetyl coenzyme A to malonyl coenzyme A. Of course, we are going to add acetyl coenzyme A, but all the acetyl coenzyme A as such will not be added. So that first of all, we activate them, where we add one carbon moiety to acetyl coenzyme A to form malonyl coenzyme A, and only after that, that malonyl coenzyme A will be subsequently added to form the fatty acid. Okay, more fatty acids are usually the even chain carbon, because two carbon atoms are added at a time. Okay. Now three, synthesis of palmitate. For the synthesis of any fatty acid, first of all, the palmitate will be synthesized, and later on, either it has to be elongated or the double bond has to be inserted. That is done after that process. But first of all, palmitate will be synthesized, and that palmitate will be released. Only after that comes the elongation and saturation. Okay. Now, brief outline of fatty acid synthesis. First, production of cytosolic acetyl coenzyme A and NADPH. Second, conversion of acetyl coenzyme A to malonyl coenzyme A. Third, synthesis of palmitate, and only after the synthesis of palmitate, either it has to be elongated or desaturation has to occur only after that stage. That's a brief outline. 4 steps we are picking. If you have to speak or write about the fatty acid synthesis, these 4 brief outlines first of all you have to give. Okay. Now we'll explain each point in detail. Now first of all, production of cytosolic acetyl coenzyme A and NADPH. Okay, production of cytosolic acetyl coenzyme A and NADPH. Okay, this figure may look very messy, as you can see, and many information you can find there, but most of them you already know. As you know that acetyl-CoA is formed in the mitochondria now, that has to be transferred to the cytoplasm. You know, pyruvate from pyruvate, with the help of pyruvate dehydrogenase, acetyl-coenzyme A would be formed, and this acetyl-coenzyme A we need in the cytoplasm, and we don't have the directly transporter of this acetyl-coenzyme A. So first of all, this acetyl-coenzyme A is combined with oxaloacetate to form citrate. This acetyl-coenzyme A is combined with oxaloacetate to form citrate with the help of enzyme citrate synthase. Now this citrate can easily go to the cytoplasm. It has a transporter. Now it can go to the cytoplasm, and after going to the cytoplasm, just the opposite reaction will occur. Just like here, citrate synthase from acetyl-coenzyme and oxaloacetate. Now after going to the cytoplasm, this citrate will be decomposed to oxaloacetate and acetyl-coenzyme, same thing. And during this process, ATP is also required. Okay, the enzyme is citrate lyase. Now oxaloacetate is produced here, and acetyl-coenzyme is produced. This acetyl-coenzyme A will go for the production of fatty acid synthesis. And we also need the HMP, we also need NADPH, and the major source of this NADPH is the HMP shunt. However, when this oxaloacetate is converted to malate and this malate is converted to pyruvate, in this process, when malate is converted to pyruvate with the help of malic enzyme, here also little amount of NADPH is produced. Main source of NADPH you can directly write from the HMP sum, and the next is you can add these two steps. If you find here there are many reactions, you can simply write citrate here, and citrate would be decomposed to give oxaloacetate and acetyl-coenzyme A, and this acetyl-coenzyme A will go for the production of fatty acids with the help of NADPH from the HMP sum. Or if you can remember the step, after citrate oxaloacetate, and this oxaloacetate to malate, and when malate is converted to pyruvate, in this step also NADPH is generated, and in this NADPH along with the NADPH from the HMP shunt is useful for fatty acid synthesis. Okay, this is the step 1, that is production of acetyl-coenzyme A and NADPH, or more appropriately production of cytosolic acetyl-coenzyme A and NADPH. Okay, this is the first step. Now the second step is the conversion of acetyl-coenzyme A to malonyl coenzyme A. Actually, in the beta oxidation, you have seen that fatty acid, when it was oxidized or when it was decomposed, then it has resulted in the number of acetyl-CoA. Here also, same acetyl-CoA molecules will be added up to form the fatty acids, that is 2 carbon, 2 carbon, 2 carbon acetyl group, but it has to be attached first of all in the form of malonyl CoA, and later on from malonyl CoA 1 carbon will be released as carbon dioxide, and same thing acetyl group will be subsequently added. But for the formation we also need the malonyl CoA. Now the second step is conversion of acetyl CoA to malonyl CoA. Okay, very simple. Acetyl coenzyme A plus carbon dioxide with the help of ATP gives malonyl coenzyme ATP. Actually, this carbon dioxide, the source of carbon dioxide is bicarbonate. This simple reaction you can remember. Acetyl coenzyme A with the help of enzyme acetyl coenzyme A carboxylase, malonyl coenzyme A is formed. Okay, if you compare the structure of this acetyl coenzyme A and this malonyl coenzyme A, only the difference is the attached CO2 part, isn't it? This attached carbon dioxide. Actually, later on, this carbon dioxide will be released, and same acetyl group will be subsequently added for the formation of fatty acids. Okay, acetyl coenzyme A with the help of acetyl coenzyme A carboxylase will give malonyl CoA. And in this process, this acetyl CoA carboxylase is the enzyme, and the coenzyme is the biotin. This biotin carries the carbon dioxide with it and transfers it to acetyl CoA, resulting in the formation of malonyl CoA. How this carbon dioxide is produced is from the bicarbonate. Okay, this is about the conversion of acetyl coenzyme to malonyl coenzyme. Are you hearing me? Hello.
Yes, sir.
Yes, sir.
Okay, now two steps are complete. First step is the production of cytosolic acetyl coenzyme A and NADPS. Second step, conversion of acetyl CoA to malonyl CoA. Now, what is the third step? What is the third step now?
Palmitate synthesis.
Palmitic acid synthesis.
Okay, synthesis of palmitic acid. Okay, this is just a few points to add on the same reaction. The ATP-dependent carboxylation provides synergy input. The carbon dioxide is lost later during condensation reactions. Acetyl coenzyme A carboxylase, which converts acetyl coenzyme A to malonyl coenzyme A, is the rate limiting step of fatty acid synthesis pathway. This acetyl coenzyme A carboxylase is the rate limiting step. That means the slowest step, and this is also the committed step. That means when malonyl coenzyme is formed, now that carbon moiety will go to the synthesis of fatty acid.
Sir,
Now the okay,
sir, during the cholesterol synthesis also acetyl coenzyme is produced in cytosol by same way.
yes, production of acetyl CoA is by the similar means. Yes, because acetyl coenzyme naturally its concentration will be higher in the mitochondria, and that mitochondrial acetyl coenzyme has to be brought to the cytosol. Actually, there you might not have studied about how that acetyl CoA is formed in the cytoplasm, but the methods is the same. Okay, now third is the reaction of fatty acid synthase complex or the synthesis of palmitate. Now the third step is the synthesis of palmitic acid, and the synthesis of palmitic acid is due to the reactions of fatty acid synthase complex. Here we have multiple steps, 7-8 steps, but all those steps are catalyzed by a single protein. This single protein has multiple enzyme activity, 7 different enzyme activity. Let's see, the fatty acid synthase complex is a polypeptide containing 7 different enzyme activity. Look, only one protein, but 7 different enzyme activity. This is, that's why in the name itself there is fatty acid synthase complex. This is the example of multifunctional enzyme. The fatty acid synthase complex is a polypeptide containing 7 different enzyme activities. Acetyl transacylase, malonyl transacylase, ketoacyl synthase, ketoacyl reductase, dehydratase, enol reductase, and thioesterase. All these activities all these activities are present on the same protein. All these enzyme activities are present on the same protein. Then the protein is known as fatty acid synthase complex. Okay, this is the general structural representation of fatty acid synthase complex. As you can see, this fatty acid synthase complex is a dimer. It consists of two similar units. This is the subunit division. However, the subunit division and the functional division are different. That's why this horizontal line you can see the subunit division, and this slanting or nearly vertical line, this is showing the functional division, because this you can see the dimer, isn't it, in the dimer, but this part and this part alone they are not active. Yes, this half part of this subunit and half part of this subunit will be active, whereas half part of this subunit and the half part of the other subunit will be active. That's why this is the functional division, but this is the subunit division. We are talking about the fatty acid synthase complex. Fatty acid synthase complex, in the name itself there is a complex, is a protein which consists of 7 different enzyme activities, and in our case it also does the activity of acyl carrier protein. It also does the function of acyl carrier protein. Fatty acid synthase complex is a protein molecule containing 7 different enzyme activities, and also in our case it also functions as the acyl carrier protein. That means in the lower organisms, that acyl carrier protein is a different protein, but in our case, the same fatty acid synthase complex acts as the acyl carrier protein. Okay. Acyl carrier protein, you can see thioesterase, ketoacyl reductase, enoyl reductase, hydratase, malonyl transacylase, acetyl transacylase, ketoacyl synthase. These are the 7 different enzyme activities. Functional division and the solvent division are different. Reactions of fatty acid synthase complex. Fatty acid synthase acts as a single unit, catalyzing all reactions. Fatty acid synthase act as a dimer with 2 identical units. Each monomer is having the activities of 7 different enzymes and acyl carrier protein. Actually, this acyl carrier protein is bound to 4-phosphopantotene. This is the protein. Okay. Now we'll go step by step of this. This protein or all the enzymes functions of the protein. 7 different enzyme functions. Okay. Now this may look very difficult when you look for the first time, but this is not that difficult. It is very easy. Now this is we are representing fatty acid synthase molecules. This is cysteine sulfidyl group, and this is 4-phosphopantotene. This represents the cell carrier protein, and this represents the main enzyme part. However, the main enzyme part as well as this acyl carrier protein is present in the same protein molecule. That's why this is the same in our case. Okay. Okay. This is the enzyme part. This is the acyl carrier part. Now, actually, we have formed malonyl coenzyme A from acetyl coenzyme A, so that we'll add subsequent malonyl coenzyme A. But for the first time, for the first reaction, acetyl coenzyme A itself comes. This acetyl coenzyme A is first of all taken by this acyl carrier protein, and it gives to this enzyme portion. Okay, this is the first reaction. Let's see, this is acetyl coenzyme A ACP transacylase. Acetyl coenzyme A acyl carrier protein transacylase. That means this acyl group is transferred to the acyl carrier protein. Now, here it is not shown, and this acyl carrier protein immediately donates it to the main enzyme portion. Now this is the first reaction catalyzed by acetyl coenzyme A acyl carrier protein transacylase. Now the second step, because we are going to synthesize the fatty acid, that is the long chain, long hydrocarbon chain, isn't it? Now one by one these units will be added. Okay, 2 carbon moiety is added here now. Now in the second step, malonyl coenzyme A will come and attaches to acyl carrier protein. This is the malonyl coenzyme A. Then malonyl coenzyme A will come, coenzyme A will be released, and malonyl group will be attached here. This is the acyl carrier protein, and the name of the enzyme is malonyl coenzyme A acyl carrier protein transacylase. Now what happens is with this enzyme 3, this is the condensing enzyme. As you can see, this is beta ketoacyl synthase. Now this acetyl group, this acetyl group will be transferred to the same acyl carrier protein. That means we are increasing the number of carbon in the chain, isn't it? Now this will go here. This is the condensing enzyme beta ketoacyl synthase. Okay, this beta ketoacyl synthase. That means now till now 1, 2, 3, 4, 4 carbons we have already assembled. But now there has to be certain reactions like reduction and dehydration that we will see. Now this with the acyl carrier protein, these 4 carbon moieties are attached, but we have to synthesize the fatty acid. Fatty acid long hydrocarbon chain and only COH group at the last. That means now we have to do something with this O, isn't it? Now first of all, with the help of enzyme ketoacyl ACP reductase, this keto group will be reduced to the OH group. And you see here, yes, reduced to the OH group. Now again with the help of next enzyme, beta hydroxyacyl ACP dehydratase. Now what happens here? Water molecule is released. This OH and hydrogen is released. That water molecule is released, resulting in the formation of double bond here. Now again with the help of this enzyme 6 enoyl ACP reductase, now this double bond here and there will be the saturation, hydrogen will be added. Now can you see it is just looking like the long hydrocarbon chain is okay. CH3, CH2, CH2, CO. Now, with the help of the same condensing enzyme, what it does is after this it is formed like the fatty acids CH3, CH2, CH2, C double bond, isn't it? Just looking like fatty acid. Now it will be transferred to the main enzyme portion. It will transfer this segment to the main enzyme portion, so that this acyl carrier protein now will again accept the malonyl coenzyme. Malonyl coenzyme it will attach, and now again this portion will come and attach here, and all the reduction dehydration process will occur, and again this will transfer to this main enzyme group. Again it will accept another malonyl group. In this way carbon chain will grow. Okay. Okay. This toolbar was this thing. Okay. Okay, we are talking about the reactions of fatty acids in this complex. You know, when you look for the first time, you don't understand it very, because there are several reactions and the lengthy reactions. But let's explain me again. This fatty acid synthase is a complex which contains 7 different enzyme activities, which is represented by the cysteine sulfidyl group here, and it also acts as acyl carrier protein. First of all, the first reaction, this acyl carrier protein accepts the acetyl group from acetyl coenzyme A, and it's transferred to the main enzyme. And in the second part, in the second step, now this will accept, this acyl carrier protein will accept malonyl coenzyme A. Only for the first time this will accept acetyl CoA, but for the other subsequent addition it will accept only the malonyl CoA. Here malonyl CoA, here acetyl CoA. Now this this acetyl CoA group will also be joined here, but there will be the release of this carbon dioxide, CO2. There will be the release of this carbon dioxide. That means earlier, when we converted acetyl CoA to malonyl CoA, carbon dioxide was added. Now the carbon dioxide is released. That means we are adding 2 carbon fragments at a time. Okay, this one. Now this, okay, this will be reduced to alcohol, as you can see, and this after reducing to alcohol, now double bond, with the removal of water molecule, double bond will be formed. Now this double bond will be saturated with hydrogen. Now it looks like we are nearly forming the fatty acid CO. This is the COH. Okay. Now this will be transferred to the main enzyme part, the cysteine group, and this acyl carrier protein will accept another molecule of malonyl group from malonyl coenzyme with the release of coenzyme. Okay. Similarly, these steps will repeat again and again, resulting in the formation of the palmitate. Okay, let's see. Following transfer of the growing fatty acid from phosphopentothiene to the condensing enzyme cysteine sulfidyl, the cycle begins again with another malonyl CoA. When the fatty acid is 16 carbon atoms long, the thioesterase domain catalyzes the hydrolysis of thioester, linking the fatty acid to phosphopentothiene. That means only when this fragment is 16 carbon long, only at that time it will be released from this enzyme portion. Till that time it will remain attached to this enzyme portion, and this enzyme will subsequently go on adding 2 carbon atoms at a time. When the fatty acid is 16 carbon atoms long, a thioesterase domain catalyzes the thioester, means as you can see, R RCOSR. This is the thioester. When the fatty acid is 16 carbon atoms long, a thioesterase domain catalyzes the hydrolysis of thioester, linking the fatty acid to 4-phosphopentothiene. 16 carbon fully saturated fatty acid palmitate is the final product of fatty acid synthase complex. All the time, only this palmitate would be synthesized and released, not the other fatty acids. If the longer fatty acids have to be formed, then there are other mechanisms. If double bond has to be formed, then there is the other mechanism of adding the double. The 16 carbon fully saturated fatty acid palmitate is the final product of fatty acid synthesis complex. Now let's see the reactions. Okay, we have synthesized palmitate. Palmitate means 16 carbon fatty acid. 16 carbon fatty acids means on the left side you have to write acetyl coenzyme A. 16 carbon fatty acid requires how many acetyl coenzyme A? It's very easy, 8 acetyl coenzyme A. And if 8 acetyl coenzyme A has condensed means how many rounds has occurred? 7 rounds has to occur. So in one round 2 NADPH, so 14 NADPH would be required, and in one round 1 ATP, that is 7 ATP are required. And on the right hand side accordingly we write NADP and ATP plus PI. Fatty acid synthesis occurs in the cytosol. Acetyl coenzyme is generated in the mitochondria, is transferred to the cytosol via a subtle mechanism involving citrate, which we have seen in the first step. That means in the brief outline, we have brief outline into 4 different steps. First one is production of acetyl coenzyme A and NADPH. Second one, conversion of acetyl CoA to malonyl CoA. Third, synthesis of fatty acid or synthesis of palmitate. Fourth one will be elongation and desaturation, which we have not studied. Okay, now to see what are the advantages of fatty acid synthesis complex. Advantages are free from interference of other cellular reactions. Just like in the case of glycolysis, let's see in the glycolysis there are 10 different steps or 10 different reactions catalyzed by different enzymes, and those different enzymes are a part of the different proteins. That means after the formation of one intermediate, now that has to travel to the other protein to form another intermediate, and it has to go to the other protein. But here this is not like that. All the enzyme activities are present on the same protein. This is one advantage of fatty acid synthesis complex. This is free from the interference of other cellular reactions, as all the reactions are catalyzed by the same protein with different enzyme activities. Next, multi-enzyme polypeptide complex is coded by the single gene. So it is easy for the synthesis of all the enzymes of fatty acid synthesis complex. If you think also, if that seven different enzymes has to be coded by 7 different genes, then the gene products to assemble and for the formation, then it will be difficult, and there will be chances that misformation will occur. But here multi-enzyme polypeptide complex is coded by the same gene, and that's why there is less chances of the malformation. So it is easy for the synthesis of all the enzymes of fatty acid synthesis complex, and no permeability barrier for various intermediates. If you see some of the biochemical pathways, just like urea cycle that occurs in the cytosol as well as in the mitochondria, some steps in the mitochondria and other steps in the cytosol. There are the permeability barriers, but here there is no problem of permeability barriers. Okay, these are the advantages of fatty acid synthesis complex. Okay, now let's see the regulation of fatty acid synthesis. How fatty acid synthesis is regulated. We have already said that acetyl coenzyme A carboxylase, which convert the acetyl coenzyme A to malonyl coenzyme A, is the regulatory step. This has the rate limiting step, that is the slowest step. This step is also the commutative step. Also, this step is the regulatory step. That is the step catalyzed by acetyl coenzyme A carboxylase. Okay, regulation of this enzyme, let's see. There is short-term regulation, long-term regulation, we'll see. And in this short-term regulation, there are allosteric regulation, and the next is phosphorylation and dephosphorylation. Allosteric regulation. Here you can see the inactive dimer enzyme undergoes allosteric activation by citrate, which causes dimers to polymerize. We are talking about the regulation of fatty acid synthesis, And regulation of fatty acid synthesis is with the help of acetyl coenzyme A carboxylase, which convert acetyl coenzyme A to malonyl coenzyme A. And in the regulation process, there are several types of regulation. First of all, short-term regulation. Short-term regulation is through allosteric regulation and also reversible phosphorylation. Talking about the allosteric regulation, this acetyl CoA carboxylase in the inactive form, it exists as the dimer, but with the help of the stimulating compound, that means the allosteric effectors like citrate, this will polymerize acetyl coenzyme A carboxylase in the polymerized form, and in this polymerized form this is active and it does its function, that is conversion of acetyl coenzyme A to malonyl coenzyme A. And if already large amount of long chain fatty acids are already synthesized, then this will inhibit the formation of polymers, hence there will be no more production of the fatty acids. Okay, regulation of acetyl coenzyme A carboxylase, short term regulation. And in short term regulation, we studied about the allosteric regulation. That is, acetyl coenzyme A carboxylase exists in the form of dimer, and when it is polymerized, just like with the inductory effect of citrate, then only it will be active. Now next is the reversible phosphorylation and dephosphorylation. This is acetyl coenzyme carboxylase, as you can see, phosphorylated form, this is inactive, and acetyl coenzyme carboxylase dephosphorylated form, this is active. If you see here, acetyl coenzyme carboxylase active, it is stimulated with the help of insulin. Insulin activates the protein phosphatase, and this protein phosphatase removes the phosphate from this acetyl coenzyme carboxylase, and when it removes this phosphate, it will be dephosphorylated, and this dephosphorylated is the active form. However, just the counter-regulatory enzymes like glucagon, epinephrine, and also AMP related kinase, this is AMPK, this all stimulates the cyclic AMP dependent protein kinase, so that this will be phosphorylated, and once it is phosphorylated, it is inactive. Dephosphorylated form is active. This dephosphorylation is brought about with the help of insulin through protein phosphatase, and the next is cyclic AMP dependent protein kinase will phosphorylate this, and this phosphorylation is brought about with the help of glucagon, epinephrine, and AMP dependent protein kinase. Okay, in short term regulation, one is the allosteric regulation, and the next one is reversible phosphorylation. This reversible phosphorylation and dephosphorylation is usually with the help of the hormones insulin, glucagon, epinephrine. Let's see the long term regulation. Okay, in long term regulation, if we take diet containing high calorie light for long time, then acetyl coenzyme A carboxylase will be induced. That is, prolonged consumption of a diet containing excess calories causes an increase in acetyl CoA carboxylase synthesis, thus increasing fatty acid synthesis. That means if you take high calorie diet, okay, resulting in the formation of fatty acid, and that fatty acid will be stored in the form of triacylglyceride. Conversely, a low calorie diet or fasting causes the reduction in fatty acid synthesis by decreasing the synthesis of acetyl coenzyme carboxylase. Now we studied about the short term regulation, that was the allosteric regulation and the hormonal regulation. Hormonal regulation is that phosphorylation and dephosphorylation, and this second is long term regulation. Other what it affects is the availability of NADPS. About 50 to 60% of the required NADPS is obtained from the HMP shunt. I have been saying that HMP shunt is the major pathway for the contribution of NADPS, and this NADPS influences fatty acid synthesis. And waste NADPS is provided by citrate molecule, as we have seen when that acetyl coenzyme A is brought from the mitochondria to the cytosol. First of all, citrate was formed, and citrate was carried to the cytoplasm, and then it was lysed, resulting in the formation of acetyl coenzyme A, an auxiliary state, and that auxiliary state through further step would also give to NADPS. Okay, the regulation of fatty acid synthesis. This regulation we have studied in multiple subcategories, that is long-term regulation, short-term regulation, and the availability of NADPH. In short-term regulation, it is about the allosteric regulation and the hormonal regulation, and the long-term regulation is if you take the low-calorie diet or high-calorie diet, accordingly this acetyl coenzyme carboxylase molecule will be induced. Now let's see the coordinated regulation of fatty acid synthesis and breakdown. That means at the same time fatty acid synthesis and breakdown will not occur. If the fatty acid synthesis and breakdown will occur at the same time, then such cycles would be known as the futile cycles, that would be of no use. But there are certain regulatory mechanisms so that certain times when we need energy, fatty acids will be broken down to give acetyl coenzyme. But when acetyl coenzyme is in excess, then among many other things, this fatty acid will also be synthesized. Now let's see the coordinated regulation of fatty acid synthesis and breakdown. Okay, this picture is also not difficult. This is very easy. Okay, from blood glucose, glucagon, this low blood glucose, then there will be the release of this glucagon, and when there will be the release of this glucagon, then this protein kinase will be activated, and this will phosphorylate this acetyl coenzyme carboxylase, and when it phosphorylates, it's inactive. And when high blood glucose means high insulin, and high insulin means phosphatase activity, then this will be in the dephosphorylated form, and in this dephosphorylated form it will be active. But here, as such, it is not always required to show all the steps. Here the major step is this acetyl CoA. With the help of acetyl CoA carboxylase, it will result in the formation of malonyl coenzyme, and in this malonyl coenzyme, after several steps, it will give rise to the fatty acids. Okay, main step here is malonyl CoA. This when the concentration of malonyl CoA is high, then this malonyl CoA will inhibit carnitine acyl transferase 1. This carnitine acyl transferase 1 is the enzyme in the beta oxidation. That means when the synthesis is occurring, synthesis of fatty acid is taking place, and there is a high concentration of malonyl CoA, that means at that time this beta oxidation will not occur. How it is regulated? With the help of carnitine acyl transferase 1. That means fatty acid activated fatty acids will not be transferred to carnitine. Once it is not transferred to carnitine, then it will not go to the mitochondria, and beta oxidation will not occur. That means this malonyl coenzyme inhibit carnitine acyl transferase 1. This is how these two processes are reciprocally regulated. That means when there is the fatty acid synthesis, then there is no beta oxidation, and when there is beta oxidation means when there is low concentration of malonyl CoA, only at that time this beta oxidation will occur. This malonyl CoA is the regulatory substance, and it regulates through carnitine acyl transferase. Okay. Now we'll move forward, and in this we'll study how longer fatty acid chain will be synthesized, and how double bonds should be inserted, and how TG is synthesized and stored. All this we will study in our next class. Okay, for today we simply synthesized palmitic acid, and we also talk about the regulation, coordinated regulation of fatty acid synthesis and breakdown. Okay, if you have any problem, if you have any questions, your mind. Okay, any questions? Okay, meanwhile I will download our attendance.
Sir, after that 16 carbon is formed, then what does that diester do? That can you explain once?
Which diester is?
After that 16 carbon is formed.
Oh, after that 16 carbon is formed. Okay, here you can see thioesterase. There is the activity of thioesterase. Then that thioesterase will separate this 16 carbon long chain fatty acid from this acyl carrier protein. And when this releases from acyl carrier protein means now fatty acid is synthesized, and that thioesterase means it will cut the bond here, isn't it? Here it will cut the bond. It will only cut the bond when 16 carbon long fatty acid is synthesized, not when the fatty acid is smaller one. It will only hydrolyze only when 16 carbon long fatty acid, only when palmitate is synthesized, it will hydrolyze. Till then, it will not hydrolyze. So, all the time, what fatty acid will be synthesized is the palmitic acid.
Okay, thank you, sir.
Okay, any more questions? Any more questions?
No, sir.
Okay. If you don't have questions, this is the end of today's class.